Hello, thank you very much for the introduction. I'm Charlotte Wright. I'm a postdoctoral researcher at the Wellcome Sanger Institute and at the University of Cambridge. And today I'm going to talk to you a little bit about the evolution of chromosome rearrangements across butterflies and moths, as well as how comparative genomics can help speciation research more broadly. And I'm very excited to be here today. So thank you very much to the organisers for inviting me. I've really enjoyed the series so far, and I'm glad to be part of it. Chromosome rearrangements are these major forms of genomic change that can come in a variety of flavors. The first is translocations, where two chromosomes will swap one part of the chromosome together. They also include fusions, where two chromosomes come together to form a single larger chromosome, and fissions, where one chromosome splits into two. And these later two types here are particularly interesting because they also alter the total number of chromosomes that an organism has, their so-called karyotype. And so these are major changes really in how the genome is organized. And perhaps therefore, unsurprisingly, they have major impacts. And so, for example, if we look at fusions, they can alter recombination rates simply by changing the total length of the chromosome, but also by modulating particularly the recombination rate within particular parts of the chromosome and bringing together alleles that previously were not interlinkage. And so this can be a powerful way that local adaptation can act, for example, in this example of the free spine stickleback. But also these large changes can have an impact on the way that the genome is folded and changes in the chromatin conformation. And it can also impact how different genes are expressed. And the simplest case to think about this is in the case of a sex autosome fusion, which can have a widespread impact on gene expression. But of course, these are also interconnected, right? Changes in how the genome is folded can have knock-on effects to how recombination is regulated and also how gene expression is. So these are all interlinked. And so, perhaps unsurprisingly as well, for a long time, the role of these rearrangements in speciation has been hypothesized, starting in the 1960s, when it was first considered that if you had two populations that differed in a chromosome rearrangement, so here population A has a fusion that population B does not, um, how this might give rise to new species. And so there's two main lines of evidence, well, two main theories. The classic theory is that when these two populations that have differential rearrangements come back into contact, they have problems in meiosis because of missegregation of these rearrangements, leading to aneuploidies. And so you have these heavily underdominant rearrangements that serve as barriers to reproduction, leading to speciation over long periods of time. But many of you that perhaps have thought about this before know that there are problems with this classic theory. The first of being why, well, if these are underdominant, then how do these rearrangements reach fixation in population A in this example in the first place? Because they're arising from a single copy and so will be present in heterozygotes from the start. And so while there have been many possible solutions proposed, such as the effect of strong bottlenecks or strong meiotic drive, these also have problems, but also things like gradual stepwise fixation of rearrangements that are perhaps more weakly impacts in fitness have also been suggested. But in general, kind of a newer line of theory has been developed that instead focuses on the effect of these rearrangements on recombination. And so this can do things like pre-existing barrier loci interlinkage and strengthen their impact, or also by altering the overall recombination rate of the chromosome, reduce the ability for there to be gene flow. And I won't talk about the evidence for these theories much more because that is what Alex will do very eloquently, very shortly. But instead, I might focus on the macroevolutionary predictions of these hypotheses. And so the most simple one of these is simply that we might expect that groups that have high rates of karyotypic variation will have higher rates of diversification because they have more chromosomal change. And so if you look in Lepidoptera, the butterflies and moths, this is what you seem to see if you look at karyotypes. So each of these dots is a genera, and you have the rate of karyotypic evolution on the x-axis and the rate of speciation on the y-axis. So it seems with karyotypes to kind of hold up. And similar patterns have been seen in other groups, for example, in spiny lizards, where a higher rate of fusions has been linked to a higher diversification rate, and also in wallabies, which I also will not speak about today because we have a Q&A with Sally Potter, who has led a lot of work in this area and might be able to give us some more insight. Okay, but so I think one other thing to keep in mind is that not all chromosomes are equal, and that can change the rate at which these rearrangements are perhaps likely to happen. And one of those things is whether they're holocentric or not. And so what I mean by this is that flies, like us, they are monocentric. That means they have a single centromere per chromosome. And therefore, we can think that if you have a fusion between two chromosomes, that will give you a single chromosome with two centromeres that could potentially be antagonistically acting. And on the flip side, if you have a fission giving rise to two chromosomes, one may get the centromere and the other will not. And so, in both of these cases, you have unstable products that are likely to be segregated in meiosis and leading to aneuploidies. And so we might expect that in monocentric systems, chromosome rearrangements are quite hard to evolve and they're very underdominant. Well, they're very hard to evolve in the first place. Whereas in holocentric species like the butterflies and moths, they have so-called centromeric activity along their entire length. And this means that if you have a fusion, the new chromosome is good to go, and vice versa. A fission, you have centromeric activity on both of those new chromosomes. And so perhaps we might think that here chromosome rearrangements are much more easy to evolve, and so we might get higher rates of karyotypic change than in monocentric groups. And just to say, I'm showing butterflies here, but they're not the only groups that have these holocentric chromosomes. A wide variety of plants and other types of invertebrates also have these types of holocentric chromosomes. But of course, this would be too simple an explanation that if you're holocentric, you have rapid chromosome rearrangements, and if you're not, you don't. And this is borne out in Lepidoptera, where actually the vast majority of species have 31 chromosomes, like you see here in Arctiacasia with the 31, and the kind of wide range of karyotypes is seen in a relative minority of species. So, for example, you have 5 chromosomes in Hyperthiosphere over here and over 200 in Polyomys atlantica. So you are still left with this kind of very paradoxical pattern of general stability in chromosome number, but when it changes, it really changes to an extreme extent. And what kind of underlies these two patterns is yet to be understood. And so, I mean, I'm a comparative genomicist, so I would argue that we need comparative genomics to try to understand this. And that's what I've been doing over the last few years. And so, with the advent of projects like Hilda mentioned, the Darwin Tree of Life and the European Reference Genome Atlas, as well as specifically for butterflies and moth psyche, we're starting to get larger numbers of genomes that allow us to directly quantify and investigate these chromosome rearrangements, going beyond what we perhaps could do with karyotypes alone. And these genomic sequences contain a lot of information about how these rearrangements may have arose and what their consequences might be on processes like speciation. And so to give you a little flavor into this, in one study during my PhD, I took the first 200 genomes that were available at the time for Lepidoptera and asked how have chromosomes evolved? And do we see the same patterns that we might have expected from that karyotype data? And so largely, yes, what we found is that around 50% of species have retained effectively the same set of chromosomes for 250 million years. And those are the ones in gray, so they haven't had a single fusion or fission. And overall, across the tree, there are around 200 fusions, whereas there were just 3 fissions. And so already we see that fusions are way easier to, or just evolve way at a much higher rate than fissions do, which is quite interesting. These are the lineages in orange. And the other thing that we saw was that actually there were less fusions than you might expect at deeper nodes in the tree. So in other words, they tend to be quite tippy, which is what you can see in the orange. And I think this is quite interesting because there's kind of probably a couple of explanations. One is that the rate of rearrangement has increased in evolutionary time, but that seems quite unlikely. Alternatively, the lineages that undergo rearrangements might actually be less likely to persist in evolutionary time. Or you may be getting reversion back to the ancestral state of unrearranged. But I think this is something that needs more data to look into more, but an interesting pattern if we're thinking about diversification impacts. What was also quite interesting that we found is that if you ask, are all chromosomes equally likely to be involved in rearrangement, then the answer is clearly not. So here we have the length of the chromosomes on the x-axis and the number of fusions on the y-axis. And you can see that the smaller the chromosome is, here I'm showing you the ancestral groups, the more likely it was involved in fusions, unless you're the sex chromosomes, in which case you always join into other chromosomes. So these are fusions between small chromosomes and the sex chromosomes, which given the disproportionate role of sex chromosomes in speciation is perhaps another thing of why we should be interested in these rearrangements in that context. Just to say, the Lepidoptera are strange because they don't just kind of preserve chromosome structure, but they also conserve gene order. So you can look across vast periods of evolutionary time, and you have gene A next to gene B, next to gene C, and so on, as shown here with the yellow underwing and the bird nest moth, where you're looking at the location of the genes compared to a butterfly, and you have this straight line indicating they're the same. And so we're kind of left with this question of why are they so stable and why do they perhaps not show the patterns that we might expect? And I think to understand this, we can perhaps look to Bateson. He was one of the obviously foundational people in genetics. And he argues that we should look at the exceptions to understand the rules. So he says, nevertheless, I may throw out a word of counsel to beginners. It is treasure your exceptions. When there are none, the work gets so dull that no one cares to carry it any further. Keep them always in covered and in sight. Exceptions are the rough brickwork of a growing building, which tells us there is more to come and where the next construction is to be. So we need to look at the outliers to understand the rules. And that's we can do this in butterflies, right? Because they have these ends of the spectrum of karyotypic diversity. And indeed, in that first 200 species that I showed you, there were roughly 8 independent origins where they've gone from having this stable structure to a highly rearranged structure. One of those is Brentfus that Alex will talk about later, but there's others too, like Lysandra and Pierid's here. And these types of rapid reorganization generally come in two flavors. They can either be purely by chromosome fissions, as you can see in Lysandra, where at the base of this genus, kind of the molecular scissors or the fission became active, and that meant that they ended up with 45 chromosomes in Lysandra belagus and 19 Corodon. And each of these blocks here, just to contextualize, is a chromosome, and the lines are the positions of genes along them. And if that gene is great, it basically means it's on the same chromosome as it was at the base of butterflies and moths, which for context, I forgot to say, is around 250 million years ago. So no fusion has happened here, just fission, which is the complete opposite to what you see in the Pierids, the cabbage whites, where at the base of this group, while fission started, fusion also started. And so this has ended up with these kind of kaleidoscope chromosomes where you have many different ancestral linkage groups per chromosome. And I think why this is worth highlighting as well is that the end output of this is that you end up with species that have 25 chromosomes and 15, which aren't so different from the ancestral state of 30, 31. And so naively using karyotypes, you might think there's been 6 fusions have happened, but actually there's been over 50 events. And so if we build models with karyotypes alone, we're vastly underestimating the total amount of chromosomal change and the models of speciation that that might fit with. So I think we can use these highly rearranged species to understand why is chromosome structure stable in most species, what causes some species to experience rapid rearrangement, and what are the consequences of them. And today I'll focus on just one of those lineages, that is Polyomatus, to start to answer these questions. And I would argue that this is a great species to, or great lineage to do this in, because they have one of the highest speciation rates known for all animals. They have around 150 species that evolved in under 5 million years. And they also have a huge karyotypic diversity from 24 to over 200. And so as a first step to understand what's going on in this group, we generated a genome for Polyomatus atlantica, which before this study was known to have a ridiculous number of chromosomes, as you can see from the cytology here. But when we generated the genome, we found it had 229. And these are tiny, tiny chromosomes which have an average length of 2 megabases, which is around a tenth of the size of a normal butterfly chromosome with just 70 genes per chromosome. And so if you're used to looking at Hi-C maps, you might think so that that's the pattern of interactions formed by physical contact of chromosomes, which is shown here on the left-hand side. You might think this is a horrible genome and you've terribly assembled it. But if you zoom in, you see that each of these tiny squares actually is a beautiful chromosome. So if you look in this little region here, you have 5 chromosomes. And this is down to the painstaking effort of Dom Absalon, who kept curating them and found every single chromosome there was. Okay, so now we have the genome, we can ask, how did it evolve? And the answer is simply fission. So if you look at Polyomatus, which is a close relative with your standard 23, 24 chromosomes, and look at a single chromosome of it, you see that it is split into 14 in Polyomatus atlantica. So these lines are showing the trajectory of individual genes and how they compare between the two. And so we've ended up with 14. The two colors are different ancestral linkage groups, and it shows that you still have very maintained gene order despite the fissions. And what we can also see, if you're wondering what these big things are at the top, that these are the sex chromosomes. And so sex chromosomes may be very likely to fuse, but they do not undergo fission. And by looking at these extremes, we can see just how true this is. At the extreme amount of fission, the sex chromosomes still remain intact. And this explains this earlier cytological observation. We can also try to ask, where do these chromosome rearrangements occur in the first place, to try to understand what potentially is the mechanism. And something interesting we found in this respect is that they tend to occur in the so-called A compartment, which is the loosely compact euchromatic component that's transcriptionally active, rather than the B component. Um, and we can also try to understand what could be the cause for this high rate of rearrangement to start with. And I think one of the clues is by looking at perhaps the telomeres, because in Lepidoptera they have a very specific typical telomere, and that is this motif of TTAGG that you get in stretchers. And so in Icarus, the unrearranged relative, you can scan along and see they have these clear lines at the end of each chromosome that is the telomere. But if you look in Atlantica, you see that peppered throughout the chromosome bodies as well are these long internal telomeric arrays, which is quite unusual and you don't see in other species. And they not only have these arrays, but they also have these retrotransposons that are specifically targeting these telomeric arrays within them, normally found in the sub-telomeric regions. So they really seem to have these kind of chunky internal telomere-like structures that can be really quite big in some cases. And so we can make wild speculations that remain speculation for now, that maybe most of the time, if you have a fission, it creates two products that are unstable because they don't have a telomere. But maybe in these lineages that have these internal telomeres, they have these nice sites that if they become cut, you give rise to kind of proto-telomeres that are stable. Or alternatively, these could be sites that facilitate rearrangements in the first place, so increasing the rate of them arising. I'm quite short on time, so I'll just very quickly say the last couple of slides, which is that I think comparative genomics is already rapidly accelerating from when I started a few years ago, and this presents new opportunities. So looking specifically at butterflies and moths here, as of 2025, we had over 1,000 genomes, which is really exciting. And so as a community of within Project Psyche, which is a project that I lead with Joanna Meyer and Mark Laxter, as well as 200 researchers in many countries, we're starting to think about how we can use this first 1,000 genomes to understand processes like speciation and diversification. And so to this end, we're using the first 1,000 genomes to look at many replicated shifts from a low rate of rearrangement to a high rate. So instead of looking at single anecdotes, we can start to see what are the consistent consequences. Is it consistently things involving telomeres that are happening or changes in meiosis, for example? And do we see a consistent impact on diversification, which is very exciting. And just to say, this is not just going to happen for butterflies and moths, and we should start to look at this in other groups. And only by looking at other groups will we really try to tease apart what is the impact of holocentric chromosomes, for example, versus more general patterns. And so as more genomes are generated for other groups, we can increasingly do that. And with that, I'll stop and apologize for maybe going over by a minute, and thank all the wonderful people that have made this possible, particularly my PhD supervisors, Mark Blackstone, Mara Lanazak, as well as Joanna Mai, who I'm now also working with, the Project Psyche community for making all these wonderful genomes possible, and all of the people that were wonderful co-authors on the work I've shown today. And for you for listening. Happy to take questions.
Thank you so much, Charlotte. That was super impressive work. So just to say, we have various options today to ask your questions to our speakers and also to Sally later. So you can just raise your hand and then ask your question. That's in some sense the preferred option. But you can also type it in the chat and I can read it out for you. Or if you prefer to ask some, very speculative question, you can put it in our anonymous Q&A option as well. And again, I can read it out. So any of those you have at your disposal. So yes, and also I would, since this first 3 sessions of the new restart of the seminar series is kind of themed by early career researchers, I would especially encourage the early career attendees today to also not hesitate to ask their questions. So with that, I'll open it to the floor. And if everyone's feeling shy this morning or is just waking up, I'll just start myself. Um, so I was curious, to what extent, I mean, maybe you hinted at this a little bit, but to what extent we can have some kind of hope of separating the rate at which fissions and fusions are occurring to their like selective advantage. And I think in the beginning you talked about that a little bit, but maybe you could elaborate a little bit on that point.
Yeah, I think one kind of easy way into kind of something for free is by looking at the genomes that are being generated now, because we have so many that if rearrangements are kind of abundant in nature and what we're seeing is them not fixing, then you might expect that we do occasionally see heterozygotes in the genomes. And I think this is kind of what we see. So in some of the lineages that are really kind of boring, they're 31 chromosomes, they do sometimes have a rearrangement, and there is, one copy has a fusion, which I think is kind of a first step to perhaps being skeptical that rearrangements occurring is the rate limiting thing, and instead it's the fixation of them. Alex is looking at me because he may disagree, but um, and yeah, and I think using PopGen data, we can start to understand what are the actual selective benefits of these rearrangements and what they might be doing.
Amazing. I'll just read one question from the chat and then we'll go on to Brent Emerson. So there's a question of whether chromosome fusion and fission are related to high frequency hybridization and introgression from Tonzu Tao.
Yeah, I think you could, well, I think you could answer that, maybe interpret that question in many ways. I think one thing that might be happening is that in these lineages that have high rates of fusions and fissions, they might be very rapidly diverging lineages that rapidly speciate. And so in these lineages, you do often get high rates of introgression because they are um, these kind of young species complexes. And so you see a bit of that. But in general, I guess what you'd expect is that if you have high rates of rearrangements with species differing by many, then you might expect very little introgression because these rearrangements are barriers to gene flow. And I think Sally Potter is an example of someone that's looked at this and tested this of if you do have complex rearrangements, you have less introgression. And it doesn't seem to be super clear cut. They still seem to be able to have some integration with them. And so it's not as simple as one or two is enough to limit this, but it depends on the system.
Amazing, thank you. So Brent Emerson,
Yeah, hi, thanks. Yeah, it was a great talk, really, really interesting. I was just wondering if you had any explanation about why we see internal telomeres in some species, what causes their evolution?
Yeah, it's a good question. I think it's all speculation.
Mm.
But what is interesting is that it's not only seen in, I showed you it in Polyomys atlantica, but in the group that that's part of, more broadly like the subfamily, there's three instances that it's where it's gone from a low to a high rate of rearrangement. And in all
Right.
three of those instances, the ones that are rapidly undergoing fission, they all have it. So it seems to be this consistent feature, at least in that clade, it's happened three times. So it's not just once, which makes it a bit more less speculative that it's
Yeah,
um,
so three times
people.
independently, you mean?
But what causes it, I'm not sure. I mean,
Yeah,
I think
okay.
it could also be a consequence of a rapid rate of fission, where if you cut a chromosome, right, and it's in the heterozygous state, you evolve telomeres at the ends. And
Yeah,
if somehow they're able to reverse that change and
yeah.
bring it back together, that's a scar effectively, right?
That's what I was wondering if that might happen. Yeah, okay.
Yeah, exactly. And I think to tease it apart, we really need to see, look at very close relatives and look at it even within some species, there's variation in rearrangement. So where there's a polymorphic fission, do you see that and try to get it kind of in action?
Yeah, yeah, great. Okay, thanks very much.
we have one more question in the chat before we'll take it to Roger. So Amin Dan is asking whether you have observed any association between chromosomal rearrangements and the ecology of the species. For example, do species living in highland environments tend to have higher or lower rates of fusion and fission?
Mm.
It's an interesting question. I'm not sure about elevation specifically, but I can see your logic and why we might expect that there could be a relationship. Um, I think people have speculated that there could be specific aspects of ecology that are related to it. So if we think the rate limiting thing is the rearrangements happening, then we might expect that individuals or species living in environments of a higher rate of mutagenic change might have higher rates of rearrangements. And so examples of that could be the pierids that feed on cyanogenic glucoside-rich plants. And so maybe this is causing these breaks in the chromosomes to happen. The other example is in the Iphamini butterflies. There they coat their sperm in toxins basically, and so maybe it's not surprising these have a high rate of rearrangements if these toxins are directly causing the gametes to undergo rearrangements. But I think these are theories at the moment that haven't been tested. I think one way of going about that is experimentally, but another nice thing about the comparative genomic approach with these many transitions, you can start to ask what are the consistent features. And so what's nice about Psyche is alongside the genomes being analysed, we have people compiling life history databases, and so we can see which of those characters might correspond onto it.
We can look forward to further insights on these questions in the future. Roger, yeah.
Yeah, I was actually just wanting to follow up on Brent's question and ask whether the internal telomeres occur in the sex chromosomes, because this lack of fission in the sex chromosomes is an interesting issue in itself.
That's a really good question. And I hadn't thought of it as a way of disentangling those hypotheses before, because they do have them. Sex chromosomes do also have them. So that could be an argument against it being a kind of a scar of fissions if really sex chromosomes do resist fissions, even in the heterozygous state.
Yeah, I mean, you can think of reasons why fission sex chromosomes might be selected against. So it's an interesting test of what Hilda was saying at the start.
Yeah, that's a very good point. Thank you.
Any more questions from anyone? Then maybe I'll just have one quick one before we move on to Alex's talk, which is whether you see any role for transposable elements in driving some of these patterns, or is there much variation in TE content across Lepidopteran genomes? Or
it's a good question, and lots of people are thinking about this. As in, I think people are along similar lines to you, and there's lots of wonderful work that's been done in polyomyceta. There wasn't kind of any striking differences in overall the repeat landscape of the ones that were highly rearranged versus not. And it can be a little bit hard to tease apart because in general, a lot of features at least in Lepidoptera, are patterned by chromosome length. So if you have radical changes in chromosome number and size, then you'll get a change in repeat landscape for that reason rather than it being the cause. But there are people doing very nice detailed studies on this, including Camille Cornet in Neuchâtel with Kai Luczek, that is looking at specifically the breakpoints and trying to understand if those breakpoints are enriched in very specific types of transposable elements and if they could be involved. So I'm excited to see what's found there.
Okay, thank you so much, Charlotte. Then we'll take it over to our next speaker, introduced by Rowan.
Great. So next up we have Dr. Alex Mackintosh. So a bit about Alex's history. He did his PhD in the Lewis Lab at the University of Edinburgh, examining how chromosomal rearrangements influence population genetics and the landscape of gene flow in Brenthus butterflies, which we're going to be hearing about today. He did a postdoc following that at Uppsala University in Martin Lascaux's lab, working on the Lewontin paradox in plants and on developing methods to jointly estimate demographic change and background selection. And he's now working at the Swedish Museum of Natural History, examining speciation in Agaranthemum. So Alex will be talking to us today about evidence for chromosomal speciation in Brenthus butterflies. Take it away, Alex.
Cool. Thanks for the introduction. Can you hear me okay and see my slides? Yeah, super. Yeah, so thank you for the kind introduction. And as you mentioned, today I'll be talking to you about some work from my PhD, where it's a project looking at chromosome rearrangements, their role in speciation, and focusing on these Brenthus fritillary butterflies. And so, fortunately, Charlotte gave a really clear introduction and overview to this idea of chromosomal speciation, where differences in chromosomes or genome arrangement between populations can promote, facilitate, or even kind of drive reproductive isolation and the emergence of new species. And so I won't give a kind of additional introduction, but what I'll do instead is give my opinion or impression of research currently on this topic. And so over the last maybe century, we've collected actually quite many examples of natural systems where we think that chromosomal rearrangements are contributing to speciation. And the strength of evidence in these different systems, it varies quite much. In some cases, we just speculate that this might be happening. In other cases, we now know quite much about what might be going on. We also have plenty of theory. So people have written whole books actually about how chromosomal speciation can happen. And so some of these models or theories are compatible with one another, others less so. And so I think the focus now as speciation researchers is to really maybe revisit quite many of these examples and ask which of these, in which cases do we find that it really is the chromosome rearrangements driving speciation, as perhaps in some of these cases, rearrangements are just coincidental with the speciation process. And an extra aim or a next step is to connect examples to theory. And so we can really work out what is the role of chromosome rearrangements in speciation. And so two traditional approaches for learning about chromosomal speciation, or kind of trying to answer these questions, are to perform crosses or to analyze quite large phylogenies with karyotype information. And so if you perform crosses in the lab, you would take individuals from either the same population or different populations or different species that differ in their genome arrangement. And then you would generate F1s, and you'd hopefully or at least attempt to measure something about reproductive isolation. So it could be the fertility of F1s, and then you could generate back crosses. You could even look at meiosis to see how rearrangements are affecting this process. And you can get really quite detailed information about what is happening. If you analyze phylogenies and the karyotypes at the tips of phylogenies, you can try to estimate the effect of chromosome evolution, so changes due to rearrangements, on diversification. And so these two approaches are quite different. They're happening on very different timescales, and I think they have different weaknesses in some ways. So if you do crosses, what you learn about is reproductive isolation today. You don't necessarily gain information about how that reproductive isolation evolved, or the processes leading up to it. And by contrast, if you analyze phylogenies and karyotype data, you actually use information from many taxa and large evolutionary timescales. So you don't have the same problem here. You're going quite far back in time, but you have little information about any given speciation event. So you can't really point at a phylogeny and say chromosomal speciation definitely happened here, and this is how it happened. You can't quite do that with this approach. And so in the talk, in my talk today, I will focus on a different approach that I think actually remedies some of these weaknesses. And so it is a population genomics approach where we're interested in this process for a pair of species where they've diverged at some point in time, and the chromosome rearrangements have happened on this same time scale. And the idea is that if genealogies also are on this same time scale, then we can use them to get information about these two processes. So how speciation has happened and how chromosome rearrangements have affected speciation. And although we can't really directly work with genealogies, I'll show in this talk that we can use patterns of sequence polymorphism from genome sequence data instead. And so maybe this is a little bit unclear at the moment, but hopefully by the end of the talk, you'll see what I mean about using genealogies, or at least genealogical thinking, to understand this process and how there's information in this data. So when I started my PhD and I joined Konrad Lose's lab or research group, I became interested in working on this pair of Brenthis fritillary butterfly species, Brenthis ino and Brenthis daphne, which have overlapping distributions in Europe. And so in the group at the time, Sam Ebden was generating estimates of species divergence for many pairs of European sister species, and actually Brenthis ino and daphne are one of the younger pairs, so they diverge maybe on the order of about 3 million generations ago. And why I became particularly interested in this pair is that there's previous cytogenetics work to show that chromosome numbers differ between individuals and between populations. So in Brenthis ino, we know that the number of chromosome pairs varies between 12 and 14, and the same is true for Brenthis daphne. And as a first project in my PhD, I generated a genome assembly for Brenthis ino, and we actually found the same evidence for variability in chromosome number. And so the individual that we high-C sequenced, it turns out that it was heterozygous for a chromosome fusion. So on one haplotype, the Z, and autosome are fused, and you see this in the high C map. But on the other haplotype, they're actually separate. And so what this told us at the time is that we have a pair of species that are fairly recently diverged, and that there's chromosome rearrangements happening at a high rate, or at least still segregating in the populations. What we didn't know at this time is whether these species have a lot of differences in their karyotypes. But we went ahead anyway and generated a genome assembly for Brenthis daphne. And when we did this, we got a really nice surprise in some ways. Given that the karyotype or the chromosome numbers are very similar, it was quite surprising. Actually, there are many rearrangements that differentiate these two genomes. So on the top, these horizontal lines correspond to Brenthis chromosomes. And on the bottom, these are Brenthis daphne chromosomes. These curved lines between them are nucleotide alignments. And these chromosomes here have no interchromosomal rearrangements. But here we have evidence for actually a history of fission and fusion rearrangements that means that we have kind of non-one-to-one syntenic relationships. And I've colored these alignments by how, at least at the time, we thought the ancestral chromosomes looked. As an example, we can follow this ancestral chromosome here, this small part that's fused to this Brenthis chromosome. And if we follow it over here, it's actually fused to a different chromosome in Brenthis daphne. And so given these rearrangements, we can ask the question, or try and ask the question of, have these fission and fusion rearrangements contributed to speciation? And I think this is a really nice system to test this question, as half the genome is essentially unaffected by these rearrangements, whereas the other half has at least some involvement in the rearrangement history. And so how we can do this is that we can start by trying to get a simplistic but useful model of the speciation history for Brenthis and daphne. And so I'll forget these rearrangements for the next 5-10 minutes, but I promise that I will come back to them. This is going back to chromosomes. But if we just focus on this population history for now, what we want to do is learn about affected population sizes, but importantly also the divergence time of these two species. If we were able to sample genealogies from these two species, and multiple lineages from each, then it'd be really easy to work out this speciation history. And the reason is that common ancestry events that are defined in this genealogy, they tell us about the rate of common ancestry through time, the rate of coalescence, and that is informative about the affected population size. And similarly, the rate or the timing of events or coalescent events between lineages, so lineages sampled from different populations, tells us about the divergence time. The same is true for gene flow. So if we had genealogies, then it'd be quite easy to identify gene flow events. We'd find common ancestry events between lineages from different populations, and these events would be before the divergence time. The problem we have, though, is that genealogies are unobservable. These are real, so genealogies are a real thing, we just can't observe them directly, and they're also quite hard to reconstruct. But what we can do instead to learn about this speciation history is that we can work with something that we can observe but relates to genealogies, and so that is genome sequence data. We can do is sample a diploid genome from Eno and a diploid genome from Daphne, or a diploid individual rather, and so we have 2 copies of the genome. And if we look in a really short short block of sequence in the genome, we can ask, are there any positions where there are sequence polymorphisms, so SNPs in other words. And if we assume that for this short block of sequence there is no recombination in the history of the sample, then each polymorphism that we observe corresponds to a mutation that has happened on a particular branch of the genealogy that underlies this variation at this bit of the genome. And so, for example, this polymorphism here is a fixed difference between these 2 species, and this tells us that the topology of the genealogy that underlies this short block of sequence must have this topology, where the first common ancestry event must be between lineages sampled from the same population. These polymorphisms, which are heterozygous sites, they don't tell us about the topology of the underlying genealogy, but they do provide information about the length of external branches. And just to be clear here, we're not trying to reconstruct genealogies, we're just trying to collect information about them. And so actually many sequence blocks will have no polymorphisms, and so there's no way we can reconstruct anything if we have no polymorphisms, but this does still provide information about the underlying genealogies, as it tells us that the coalescent history concludes or finishes before there's been time for any mutations to accumulate. And so we can do this along the genome many times, and we can collect 80 million of these short sequence blocks, and we can tally how many times we see one of 256 different possible mutation configurations. And this number of 256 comes from the fact that we only count certain mutation types up to a count of 2 or more, and then we stop counting. And then what we want to do is use this data to estimate the parameters of this model. And so the split time, these affected population sizes, and the rate of gene flow from one population to another, which is we can use this term or parameter, the effective rate of migration, to describe. And we can do this calculation using a generating function approach developed by Conrad some years ago, but has been extended and made more efficient in recent years. And so we can calculate the we can calculate the likelihood of our model parameters given our data, which is these mutation configurations, or integrating over the possible genealogies that are consistent with the polymorphisms we see in blocks. And when we do this, we estimate this speciation history, where the two populations split just over 2 million generations ago, and we have large effective population sizes on the order of 100,000 or even 1 million individuals, effective individuals. Importantly, we also estimate a non-zero rate of gene flow from Eno to Daphne forwards in time. And this rate of gene flow, or effective migration, corresponds to just 1 migrant every 16 generations, which is really quite low, but I convinced myself during this project this gene flow is real. And so I don't have time to share evidence for this, but if you're interested, you can ask me later. And so now after we've dealt with all of this modeling, we can return to these chromosomes, and we can use this modeling approach to learn about the history of gene flow on each one of these chromosomes. And so we can essentially just fit the model with the one tweak we do is that we fix this split time. We say the split time is a real event that has happened in time, but these other parameters, they actually correspond or try and kind of emulate processes that can vary along the genome. And so background selection will mean that the NE parameters will vary chromosome to chromosome or locus to locus. And similarly, if there is variance in selection against migration, we also expect this ME parameter, effective migration, to vary on different chromosomes and different parts of the genome. And so we can do this first for these non-rearranged chromosomes. And when we do so, we find that most of these have quite a similar rate of effective migration since the onset of speciation. But one of these is a real exception, and reassuringly, we have an explanation for why. This is the Z sex chromosome, and given hemizygosity for this sex chromosome, we expect that there should be stronger selection against migrant ancestry on this chromosome. So in a way, this is, well, it's both interesting, we've learned something about the biology of these species, but it's also reassuring that we find a lower effective rate of migration. And now we can do the same for these rearranged chromosomes. And what we find is that although there is some variance between them, actually all of these rearranged autosomes have a lower rate of effective migration than when compared to these non-rearranged ones here. And I don't quite know why there is such variance here, but my kind of best guess is that actually the timing of different rearrangements relative to the speciation process will affect just how much this effective migration rate is reduced if these are barriers to gene flow. Because this likelihood calculation is really efficient, we can actually extend this and go along the genome and fit this model and estimate the effective rate of migration. Here I'm showing an example for Brenthisino chromosome 8. And so this chromosome is made up of 2 ancestral chromosomes that fuse together at some time, and the fusion point is just here. And so if we look along this genome, we'll see that effective rate of migration, our estimates vary, but actually surrounding this fusion point, we have a valley of very low gene flow. And this is what we would expect if rearrangements are perhaps selected against. And so if you were a neutral locus here and you move into the Daphne population, but you're carrying this arrangement, then you will only be able to introgress in a way if you can recombine away from this rearrangement. It is worth noting that these estimates of ME will have some uncertainty associated with them because we're fitting this model in just smaller parts of the genome. And so we can summarize the results in an alternative way that avoids this issue to some extent, where instead of looking at the estimate of one parameter, we can instead look at the statistical support in terms of log composite likelihood for two different models. One is where the effective rate of migration is 0, so there's no gene flow in this part of the genome since the onset of divergence, and an alternative, which is the background rate of gene flow, so the genome-wide estimate. And so for each window, if you're above this line, then you have more support for this model, like a barrier, essentially a complete barrier to gene flow, than a non-barrier window. And what we find is that we do see this here and here and here, and actually just right next to our rearrangement point on this chromosome, we find a small mountain of composite likelihood. And actually this point just here is the window that has the strongest statistical support for a barrier to gene flow in the whole data set. And if we go along the whole genome, we find that a large minority of windows have a support greater than 0, so favor this model. And so we could say that 23% of the genome, we have evidence that they are barriers to gene flow. We can do the same, but for non-rearranged and rearranged autosomes, and we find that windows are much more frequently categorized as barrier windows on rearranged autosomes than non-rearranged ones, and this is a statistically significant difference. The same is true for the Z chromosome, and actually we get an even stronger effect if we limit our sampling of windows to just 1 megabase either side of a rearrangement point like this. And so I'll conclude by returning to these two kind of questions or aims that I mentioned at the start of the talk. And so, at least my opinion, is that what we want to do is identify cases where chromosome rearrangements have promoted speciation. And then we also want to connect these examples to theory. And I think in this work, we've done this first aim quite convincingly, or at least I have convinced myself that chromosome rearrangements for this pair of Brenthus butterfly species, they really have promoted speciation. They're surely not the only barrier to gene flow. They are not the only process or thing that is driving speciation. That can't be true, but it does seem like they are important. This second aim, to connect this example to theory, I think we actually haven't quite managed to do it, but we haven't managed to do it for maybe an interesting reason. The approach we have taken is to estimate the effective rate of migration along the genome. So we've estimated this parameter, but for two loci, a neutral locus and a selected locus, the effective migration rate of our neutral locus is dependent on the actual migration rate, and then the rate of recombination relative to selection and recombination. And so this, in a way, is really what we're interested in. We'd like to know about just how strong selection is against rearrangements. We'd also want to know about whether rearrangements modify recombination. But because we haven't fit a model that actually includes selection or recombination, we've just fit a demographic model to estimate ME, we haven't quite managed to do this. And so I think, in a way, I favor the idea, or at least if I had to bet, I think that these rearrangements probably do have there probably is some selection against them, but an alternative is actually rearrangements are modulating ME via recombination. And it doesn't have to just be that recombination is reduced in the Brenthus Daphne genome, the population where gene flow is incoming. It could be that in hybrid individuals, recombination is reduced due to these rearrangements. And so I think connecting empirical examples to theory is still quite challenging, And I think one needs to fit really explicit mechanistic models to do this, which is exciting but difficult. And so I'd like to finish by thanking my PhD supervisors, Konrad Loser and Simon Martin, as well as the other members of the Loser Labs, Dom Lech, Derek Setter, Gert-Jan and Sam, as well as our collaborator in Barcelona, Roger Villa. I'd like to, thank you all for listening. And that's all.
Thanks, Alex, for the amazing talk. Thank you. So I'd like to open the room up for questions, first from early career researchers, if we have any questions initially coming from people at an early stage of their career. No? Do we have any questions from anyone else? Katie?
Yeah, thanks for this really nice talk. It was super cool. And your point when you showed like the rates of gene flow on the different rearranged chromosomes. So there was the orange one that had higher gene flow, but it's also probably the least rearranged chromosome. So I wonder if it might not be age, but just it can still sort of do what it needs to do because most of the chromosome is collinear.
Yep, I totally agree. I think that at least, when I was doing this work, it was quite hard for me to imagine what does meiosis look like in an early generation hybrid. And so clearly there's going to be multivalence and problems, but it won't all be equal. And so I think you're right that maybe, for example, this chromosome 5, this is a chromosome that is better able to evade problems of meiosis.
And could I just, a quick technical question? So when you did the estimates of your demographic parameters, was that using the whole genome or did you just just use the non-rearranged chromosomes?
It was using the whole
genome,
yes, everything together. One could argue that you would ideally want to jointly estimate the variance in ME along the genome as well as the split time, but that is a little bit tricky, and so instead we do a two-step procedure.
Okay. Great, thank you.
Great, thanks. There's a question from Stuart.
Can you hear me?
Yes.
Hi, Alex. Lovely talk. Hello. Can you go to your barrier support diagram? Yep. So you've got your peak barrier support there, right bang where you would expect it. And then relatively close, you've got almost the strongest signal for gene flow. It's not the strongest, but it's almost. And that's a pattern that I see in looking at gene flow across hybridomes, that often you have a coupling between barriers and right next a high gene flow area. And I wonder, is this compensation for the barrier mechanism? So I love seeing that here in your talk. And I just wondered, have you seen this in other systems you've looked at, or has anybody else? I just find that pattern very striking.
I've not seen it elsewhere, and I haven't really thought about it. But I think what it does, if I think about it right now, I think it does make me think, say we decide we want to really estimate this, and we fit a model like that in Ashback 2017. So we have a recombination map, and we have, we're trying to estimate the density of selected regions. I don't know if that, that's still assuming that selected sites are all independent of one another, and you would, you sum over them. I don't think that would give you any kind of dependence or recombination interference. So I think that maybe this is a hard thing to get at, or I don't even know how to approach the problem.
So
but it's an interesting idea.
I had the second question, which is also kind of bizarre, which is, so you just flicked to the Barton-Bengtson And do you think it's useful to think of the effective recombination rate?
I think it is to some extent. So you can imagine. I feel like this is convincing. Like this plot here, where we have ME along this chromosome. But I think, to really interrogate one's results, what you'd ideally like to do is, if you have a recombination map, you fit a maximum likelihood model where you condition on the recombination map, and your data is maybe your ME estimates from Gimbal, and then the one parameter you'd estimate is the density of selected sites under a really boring model of just polygenic selection. So all sites in the genome have some probability of there being selection against migration. And then maybe that landscape, maybe it looks like this. And that, that should be really alarming. You should then note that actually this signal is just driven by variation and recombination under a really boring but realistic model. And so maybe considering recombination explicitly isn't a great way to find barrier loci, but it's a good way to check your confidence as to what is really driving that barrier effect, like a good null model, in other words.
Thank you.
Great, thanks. We have a few questions in the chat. I think we might only have time for one or two more. So first is from Tongjo Tao, who says, "Hi Alex, thanks for your fantastic talk. I have a slightly layman's question. After a gene flow barrier caused by chromosomal rearrangement, does it lead to a reduction in the effective population size of the populations, the butterflies?"
I think not. I don't think so. I think that in terms of diversity, if you measure nucleotide diversity, then once you shut off gene flow, then you have lower diversity. But if you fit a model like this, then you should account for that. That actually what you partition, the processes of just coalescence in one population, and then you include, well, how often actually does a lineage leave to another population, and then they don't come back together. And so I think it wouldn't affect NE, but if you just measure nucleotide diversity or you run PSMC, then absolutely this is affected by gene flow. But I guess that's NE as a measure or an idea.
Great, thanks. And I can pass over to Hilda if you think we can. Yes, so we can move on to the Q&A session now.
Great, thank you so much, Alex. And maybe you can still have a brief look at the chats for the ongoing discussion about some of the other points. I'll just stop the recording here.